I'm Genevieve Anderegg at the
state of fluid preserved insects in United States and in large collections.
Denver Museum of Nature and Science, and this is a survey of the current state of fluid preserved insects in U.S. entomology collections.
It wasn't going here, right? Okay.
Hello, I'm Genevieve Anderegg at the Denver Museum of Nature and Science, and this is a survey of the current state of fluid preserved insects in U.S. entomology collections. Fluid preservation has been used for several centuries, but is most common with vertebrate taxa, including fish and herpetological specimens. As we all know, insects are most commonly preserved pens. However, insects can be preserved in fluid as well. Fluid preserved insects can be valuable scientific resources as long as they are properly preserved. However, the best practices for managing these specimens can be less established than other more common methods of entomology specimen preservation, such as penning. Fluid preservation serves many needs within endo collections. It is ideal for certain particular categories of specimens, such as immature life stages, aquatic species, and soft-bodied adults. It can also serve as an intermediate step between collection and penning, such as when large amounts of specimens are collected with bulk catch methods. DNA can also be sequenced from fluid preserved specimens and also directly from preservative ethanol, highlighting the future use of these fluid preserved specimens. However, fluid preservation's biggest challenge is the continued upkeep these specimens require. Fluids tend to evaporate, which can expose specimens to deterioration and requires constant management in addition to new fluid or topping off to ameliorate. Another complication is that some of our most common fluid preservatives, alcohols, also tend to decrease in concentration over time as they evaporate. This is because alcohol preservatives are often mixed with water to give an intermediate concentration, such as 70%. And when that mixture experiences evaporation, it is mostly ethanol evaporating, which in turn lowers the concentration of the fluid below ideal preservative levels. For example, a container initially filled with 70% ethanol will have a concentration lower than 70% if any fluid has evaporated. Topping off or refilling a container with more fluid during routine collection upkeep can be a complicated task. If a collections manager wanted to achieve a specific fluid concentration of topping off, each vessel would require a calculation to obtain the specific ethanol concentration used. That concentration depends on the measurement of the exact concentration and volume of the remaining fluid commitment for remediating. This process is often time-consuming and technically challenging, and is often not possible for most institutions. Topping up practices also depend on other storage factors, which impact rates of evaporation and concentration decrease. These include the container enclosure material, size of the container, the amount of specimens, and the kind of specimen. Exactly how these influence evaporation rates is not thoroughly studied in entomology collections. I was interested in what collections management practices were being used in entomology collections across the U.S. for fluid preserved specimens. The goal was to determine where many collections were starting from, and then to give recommendations on areas for improvement. First, I conducted a nationwide survey of the state of fluid preserved insects. Then, in order to gather useful management techniques and information that entomology collections might implement, I interviewed several experts in entomology collections and fluid preservation in general to learn how they manage their collections, what techniques they found useful, and what they recommend. First, the survey. In order to assess the state of fluid preserved specimens in U.S. entomology collections, I asked respondents about the storage state of fluid preserved specimens, what management practices they use, and the difficult safety experience. I identified 243 U.S. entomology collections following a list from COPET all 2019, and I distributed the survey using direct emails to the ECN listserv and the ECN annual meeting this year. I asked respondents about their job title and institution information, and about the specimens they store, the amount of taxa and life stages to store fluid, as well as the containers and closures they use, what sizes and materials the containers are, and what specific fluid concentration. I also asked about management practices, such as how they assess fluid concentration, how often they do that, and how they do that, and also how often they top off containers. I also asked if they still food preserved specimen records in their database. Overall, 84 institutions consented to the survey, and 81 of those respondents reported that they had food preserved insects in their collections. Of the 81, 6 included detailed information about the composition and state of food preserved insects in their institution. Overall, the most common respondents were collections managers and curators, but I also had entomology professors answer who oversee bee collections, museum directors, and a variety of other entomology collection staff. I received responses from collection institutes, colleges, and universities most frequently, followed by public museums, government research labs, nonprofit organizations, government agencies, including state governments, and one private collector. I also received responses from institutions of all sizes, including over 3 million insect specimens to under 1,000 insect specimens. Every insect taxonomic order was reported to be served with fluid with a minimum of 6 institutions per Megaloptera, a maximum of 44 per Diptera. Orders with specific life stages that are preferentially kept with fluid were well represented, such as Odonata and Ephemeroptera, as well as groups that are historically and commonly distinct, Diptera and Coleoptera. All life stages were represented as well, with a minimum of 22 reported that they store eggs in fluid, and a maximum of 3 respondents saying that they store adults. Survey responses show that entomology collections store their food preserved specimens in a wide variety of container sizes, types, and materials. Containers tend to be smaller, with all 58 responses to the question reporting that they have containers smaller than 1 liter eggs. Most common sizes reported were 2 grams and 4 grams. Glass was the most common container material reported, followed by polypropylene plastic, then polyethylene plastic, and a variety of other plastics. Plastic polyethylene caps were the most common container closures reported. Multiple stoppers materials were widely used in several collections, including neoprene, silicone, and cork. Several respondents attributed a large variety of storage materials to the nature of recently building food preserved specimens in different containers. Ethanol was the most common food preservative, followed by glycerin, isopropyl alcohol, formalin, propylene glycol, Carnegie fluid, and barber's fluid. Ethanol was most frequently used at 70% concentration, followed by 95 and 80% concentration. Respondents were also asked if their collections stored to be bulk and sorted specimen lots, such as samples from pitfall and tracks. Out of 55 collections, 36 maintained bulk samples, 18 do not, and 1 respondent was unsure. Bulk samples were reported to be in various stages of active sorting in 7 collections. Additionally, 1 collection reported that they were actively disposing of bulk blocks due to space and time considerations. When asked how often containers in a collection are visually checked for low preservation levels, the majority of respondents reported checking once a year or not at all. Once every 5 years, once every 2 years, and once every 2 to 6 months were less frequent survey responses, and these were usually in smaller collections. The timing of topping up practices is similar to visual monitoring schedules. 13 collections reported topping up containers never or very infrequently, and 12 reported topping up once a year. Additionally, 3 collections top up containers as needed or as most. A wide variety of topping up practices were also reported. 25 top off with the same concentration as the original fluid, 70%. 12 collections top off with a higher concentration than desired because they assumed that some of the concentration is lower due to evaporation. Instead of topping up, 2 collections replaced fluid completely, and only 1 collection used the hydrometer to measure the concentration of each container and then calculate the exact percentage of ethanol to add in order to maintain a specific concentration. Due to lack of time, 1 collection does not top off at all. The most common difficulties experienced were lack of time and lack of funding. Food preserved specimens were also not authority for 16 institutions, and 15 institutions said that the collection did not have established procedures and best practices of how to monitor food collections were unestablished, which created difficulties in managing these collections. 38 institutions reported storing records of food preserved specimens in collections management systems, with scan and specify being the most common, followed by Excel spreadsheets. However, only 5 of the 53 respondents reported that their collections used their collections management systems to track the conservation and replenishing preservative fluids. The themes of the survey is that entomology collections in the U.S. have a wide variety of insect specimens preserved in fluid overall, taxa, life stages, and many collection methods. There's a wide variety of storage states reported, including container size, closure, material. Topping up frequency and methods are likely exposing specimens to less than ideal preservation conditions in many collections, and the lack of record keeping for fluid specimens is a barrier. Next is the expert interviews. These were 60 to 90 minutes in virtual, and I gathered advice and experience from these interviews. I interviewed Johnston, who's the general food preservation expert, Dr. Oboisky from the Essex Museum, Chris Bridger from the Cal Academy, Dr. Young from the WIRC, Jeff Stevenson from DMNS, and Dr. Duvall from INHS. For a first recommendation based off of the survey and my interviews with experts is more frequent and intentional measuring and remediation of food levels. While measuring food constitution is a difficult process, establishing a profile of the preservation state of fluid preserved specimens can help managers determine what steps need to be taken in the collection in order to maintain ideal food constitution and preservation state. Establishing a schedule for topping off that is informed by the size, closure types, and fullness of containers, as well as current food concentrations, will help avoid management techniques that might accidentally result in concentrations that are too high or too low for our business. I also recommend balancing preservation and accessibility in food storage methods. Specimen storage strategies always represent the push and pull between balancing preservation and accessibility. Smaller bottles are easy to work with, but are harder to assess preservative concentration and more time consuming to top up. They're also more susceptible to changes in storage and temperature and relative humidity. Large jars with small vials inside lowers the specimen to fluid ratio, which gives collection staff more time to remediate fluid levels before specimens are exposed to decay. However, specimens are harder to access in these type of jars. I also recommend increased cataloging of fluid specimens and preservation state tracking. Overall, the storage state of fluid preserved specimens is not well tracked across taxol, including within. Tracking preservation state, including what fluid and concentrations are used, is essential for the application of management cuisines, especially with the variety of specialized fluids used for collection, euthanasia, fixation, and preservation, and enzymatic departments. While fluid preserved specimens are not cataloged as frequently as food specimens, it is essential that we catalog these specimens because they contain, they represent taxol like in collecting methods that are not as well represented in food specimens, but are still. Overall, entomology fluid collections contain a wealth of biodiversity data, but are largely undermanaged due to a lack of useful management recommendations and collection resources. Fluids preserved specimens and endo collection in the United States are diverse in type, taxon state, storage state, container size, material, levels of sorting, and management practices, fluid level, concentration measuring, and remediation. These specimens likely contain a wealth of valuable biodiversity data and a focus on proper storage, food collections management, and record keeping to keep these specimens visible to science and make them available for research now and for long into the future. Thank you to everyone who took the time to answer my survey. Thank you to my advisor, Dr. Christine McCain, and to Emily Baker for introducing me to food preservation. Here my research. Thank you.
We have time for one short question. If you can speak in just speak.
Yeah, I'm one of these museum curator, manager, and technician, everything in one person. So we don't check. I don't check the alcohol connection every year. I just don't have time. So what I started doing and is to I'm topping the ethanol and adding about 5% glycerin. So I'm topping with ethanol mixed with glycerin. So if it evaporates, the glycerin keeps the insects from dying. So I just would like, if come up with what you think that was a solution for not being able to monitor concentrate.
So
propylene glycol.
Let me ask real quick. Genevieve, you're on the Zoom call here. Everyone should be able to hear you. There's a question and comment about using glycerin in ethanol and also propylene glycol. Do you have any information on that from your study?
Yeah, so checking every single concentration in every vial is very unrealistic for many collections, and often finding a solution that you can kind of apply to every vial, like topping off with an intermediate concentration, like a little bit higher than what you're looking for in your preservative solution to try and combat that effective evaporation. That can work as well. Someone mentioned that other people have used things like glycerin to add to the vials so that once the evaporation happens, the specimen has a kind of coating around it that is protected from desiccation. What I would recommend is, if it's possible, is try and just get an overall sense of your fluid collections and what is kind of happening in them, like get a sense of what is the overall evaporation by tracking how evaporation is happening over time, so that you just get an overall feel for what's happening in your collection. And then you can apply that one method to many different vials if necessary. So do a little bit of work and a bit of a survey first, and then figure out what the needs of your collection are. But those methods are totally valid and often what's only possible for most collections.
Great, thanks, Genevieve.
All right, next up, Andrew Johnston, and he's going to talk about managing entomological collections for non-traditional users.
All right, thanks. So today I want to talk a little bit about some of the lessons we've learned with the NEON collections in reaching out to non-traditional collections users. So NEON, or the National Ecological Observatory Network, is a continental scale program funded by the NSF to monitor and forecast ecological change. And so NEON collects standardized organismal and environmental data for the next 30 years. And these data are collected across 81 sites, both terrestrial and freshwater aquatic sites in the continental United States, Alaska, Hawaii, and Puerto Rico. And from these collections, 181 data products are freely and openly available to the research community. They're available through the main NEON data portal and also our biorepository data portal. So this is a map of all the sites where collections happen, and any physical sample collected at these sites makes its way to us at the NEON Biorepository at Arizona State University, where we're tasked with preserving them and getting them out as loans to the research community. We receive somewhere around over 100,000 samples per year from a diverse set of taxa, and they're stored under a lot of different types of collections management strategies. So, some look like traditional natural history collections, pinned insects, pressed plants on herbarium sheets. We also have a lot of tissue samples, DNA extracts. We have bulk samples that have no taxonomic identifications. We also have soils and other environmental samples as well. So I want to talk for a minute about curatorial strategies. How do we manage the things in our collection? So here we have a drawer that we conveniently left some expansion space in, and we want to incorporate some new material into our collection. So of course, this is something that we all kind of think about and are familiar with. Maybe we curate by curatorial event, a donation comes in, or an identification event. Maybe an expert comes and identifies some of our material. We slot it into the expansion space, and that's great. But what happens if we want to incorporate that new material by geography or date collected? Obviously, that gets a lot more complex. So of course, we all know that the more curatorial levels, the more granular you get with this, that compounds the sites of possible expansion within your collection. So then we also need to think about, well, how are these specimens actually going to be requested? And so these are all examples of how specimens have been requested from the biorepository collections we manage. So we have our traditional request, can I have all of a particular species? That's pretty easy. You pull that species out of the collection, send it off as a loan. Doesn't matter where or when they were collected. But then we also get a lot of requests for things. We want everything from a certain site. Maybe we want to look at population dynamics through time, right? So of course, now we have to look for things within taxa and also pull out things by geography and location. We also get a lot of requests for things from a particular time range. We don't care when it's collected. We want every single beetle from 2018 so that we can measure functional traits across the US, right? And so, okay, that gets even more complex. But the hardest to fulfill are what I would call randomized subsampling events. And so this might be a researcher is interested in seeing every voucher related to certain haplotypes from associated DNA barcodes. Or perhaps a researcher is interested in a huge scale across time and space, and they have to subsample those in order to complete their research task. And so these are all loan requests that we've gotten. And so then, of course, we know our curation has to reflect specimen use of that collection. So this is a picture of a typical drawer in the NEON Biorepository with a header label that we would put here. And like most collections, we start taxonomically. We put our collections by taxon. We have a scientific name there. We also cite the source for the nomenclature. This is different from the source that was used for the identification resource, but we think that curating is a scholarly act, and we want to track that and the resources we use to get those names into our collections. For us, then, we also annotate these by the year they were collected and which NEON domain site they were from. So in a single unit tray, you have this unique combination of year and domain. And then we orient our specimens numerically within that unit tray so we can go and pull out taxa very quickly. The final thing that we try to annotate on our labels is whether this is an expert ID or not. And this is to say that all of our insects are first identified by parataxonomists, field scientists that work for NEON, and then a large subset go to experts to confirm or further refine those identifications. So we want all of that to be visually available as you're browsing our collections. Of course, there are some trade-offs for this approach, because the first is space. So if you have a species like this is all one species that is encountered regularly, but in low numbers. That means you have a new unit tray for every specimen you get every year, right? So that's definitely a trade-off, but there's also labor trade-offs there, because those are more header labels to print out, those are more unit trays to move around, and our collections workers need to be really diligent when they're accessioning new material and making sure they follow all of these levels of curation. We also have a large number of bulk samples. So when we curate our bulk samples, we have some 40,000 pitfall samples that are from structured sampling. And so first we arrange them by site. So there are in this example, there are 47 terrestrial sites where pitfall traps happen. And each one of those sites has 10 plots. So we have 470 sets of pitfalls that go on for 30 years. And within that grouping of samples, we arrange them chronologically, and we annotate the start and end year of that particular unit tray within our collection and arrange them on the shelves. Okay, so now I want to think about navigating collections. So that's a little bit of how we've kind of put things there, but how do people get around our collection? And so this looks like most of the collections represented here today. We have labels on the outside of our cabinets and 2 labels on our drawers. We have a higher taxonomy down to species level. Our families and subfamilies are in phylogenetic order, while tribe, genera, and species are alphabetical. Okay, so with that information, most people in this room, with a quick little lay of the land in our collections, you could find the taxon that you're looking for in our collection. But what if your users are not systematists or regular collection workers? And that's the case for us. Our visitors, the people requesting loans, and frankly, most of our collection workers who are largely undergraduate students who maximally have 1 entomology class under their belt, how do they get around these collections? And so the first thing we tried doing, I printed out these hard copy lookups. These are alphabetical carabid genera that tell you the tribe and the subfamily. And we use these little magnetic holders to put them on the outside of the cabinet. Well, that's great. And that can help you kind of carry these around with you and find places to put beetles away if you're working in our collection. But then I was really thinking, how can we bring digitized collections data back into our physical collection spaces? And so what we now have on our cabinets are little tags that look like this. There's a QR code that you can use with your cell phone camera, or there's a little NFC tag. There's a little microchip, the same technology you use for Apple Pay, Google Pay, things like that. You tap your smartphone to it, and it'll give you a little notification. Do you want to go to this website? If you say yes, we have a simple taxon lookup where you can start typing in the taxon, and dynamically, as you type, it's going to narrow down your results till you get what you need. And within that, it's going to tell you the cabinet number, or maybe NA, because we don't have that taxon in the collection. It's also going to give you information on subgenus, which is important because that's a level below my genus level lookups that we were able to put on the hard copy printouts. We have an equivalent lookup for our pinned mosquito collection, which is also very important because less speciose than carabids. Mosquitoes are almost all in one genus, and you have to really know your subgenera there. So this is the first step, but I'm imagining next is to put these tags on all of our cabinets and think about having dynamic maps. So if you tap one of these tags, it will dynamically pull up all the digitized georeference specimens from your cabinet by what family's in there, right? Or your collection workers, if you're doing inventories, can you get a specimen count, can you get a species list of what should be in that collection dynamically, and thinking about all of the outreach and education benefits that might be there. And so all of this is pretty straightforward and simple. It's a 60-line R script that is really powered by a simple spreadsheet which includes taxa and your cabinet level inventory, which could be to any level, it could be to family, genus, species, whatever you've inventoried your collections to. So finally, I just wanted to kind of give a quick note on some thoughts on interfacing our collections with, well, external research groups. In my case, this is macrosystems ecologists, right? A very non-traditional group of people in collections. And so we're in a really great situation here with the Neon Biorepository. We're living in the expanded specimen paradigm. Our specimens are all born digital. They have extensively linked metadata. They come to us when the specimens arrive, we can populate our database with everything. These specimens are linked to other samples, other types of data, DNA, barcodes, and DNA extractions, et cetera. So this is really cool. This is really exciting to kind of be there and be thinking about these things. But what I would say is that every time I get a loan request, I have to use custom SQL queries on our database or download data and use R scripts to parse them to be able to figure out if we even have the taxa that these ecologists are interested in, that need to answer their questions. And so I think that as we move forward, if we want to reach out to these groups, we need to continue to teach them Darwin Core standards and teach them about our data culture and how we manage our collections. But also, as we get more data, we also really need to develop informatics skills and tools for ourselves to even be able to serve these other communities. With that, I'd just like to thank the team that I work with at ASU. If you're interested in any of our specimens or data, I can talk to me or email us at biorepo@asu.edu. If we have time for questions, I'll answer them, but this QR code will take you to the GitHub repository with that lookup that you could try to adapt for your collections. Thank you.
We have time for one question.
Yeah.
Because this data is more digital now, more and more younger accepting loans from museums that are based on specific barcodes within those collections. And I was wondering if you've noticed that, or if you want to comment on it, because like thousands of specimens with very specific barcodes.
Yeah, so the question comment was kind of about that now a lot of loans are maybe being happened by specific barcode barcode identifiers. And yeah, so we're seeing that for users who know how to use our data and our data portals, we get very specific requests and we'll get a list of, hey, pull these 100 specimens, and then you have to be able to find those, right? But for me, sometimes the harder part is like, do you have enough specimens that I could answer this big question? And I have to figure out which specimens and then be able to also find them. So yeah, it's great that we're cataloging all these things and digitizing them, but being able to find them efficiently is very important too. They're visible. Yeah, they're on the top. They're not on the bottom. Okay. Yeah. Thank you. Yep.
Next up is Jeremy Frank from Hawaii, and he's going to talk about the digitization of the Hawaiian cave arthropod collection.
All right, hello all, and greetings from sunny Hawaii. There we go. So greetings from sunny Hawaii. It's certainly very different from here, but I'm glad to be here and talk a little bit about the things that can be found there. So I'd like to start off by talking about one of the things that, of course, everybody imagines when going to Hawaii, which would be the Hawaii State Code governing biological surveys. So there's a lot of legalese in there, but really what this says is that the Bishop Museum is tasked to maintain a collection of every species found in the Hawaiian Islands, which is really cool that we are enshrined in state law as having that responsibility. And it's a significant responsibility because Hawaiian Islands biodiversity is really imperiled. And can talk a little bit about some of the things that the entomologists might be actually imagining when they're going to Hawaii. Things like our yellow-faced bees or our Drosophila, these really cool lineages, which are super diverse in Hawaii. But today I'm going to be talking a little bit about some of the things that maybe are not quite so commonly known. are Hawaiian cave arthropods. And in fact, for quite a long time, they were not known at all. It was thought in the world of cave biology for a long time that Hawaii did not have endemic cave adapted life. And there's a couple of reasons for that. One is that on a geologic time scale, Hawaiian lava tubes are relatively young, and it was thought that there just wasn't enough time since formation for cave adapted life to evolve. Another reason is that typical cave resources that are common in mainland caves, limestone environments, are not found in Hawaiian lava tubes. We have one bat, I believe, in Hawaii, but I'm not really a vertebrate person, so be careful quoting me on that. So there's not really guano as a sort of base resource for things to feed on in Hawaii. Another major resource for mainland caves is rivers, so water bringing in nutrients. But lava tubes are very different environments. They're quite fragile. So if a river runs through a lava tube, it's not bringing in resources so much as it is sending on a path towards imminent collapse and destroying the whole thing. And finally, the last reason that Hawaiian lava tubes weren't supposed to have these sort of cave adapted animals is that the predecessors, which were well adapted to evolve into cave forms on the mainland, are not really found in Hawaii. As you can probably imagine, the sort of pre-adaptations that make something very good at moving into a cave and never coming back out are not really so good for moving across the ocean and settling on an island. But as it turns out, during a survey in 1971 by Bishop Museum scientist Frank Howarth, 2 cave-adapted insects were found in these lava tubes. So a plant hopper and a cricket, which were new to science and quite obviously, as you can tell by their lovely pale coloration, true cave-adapted life. So how are these things surviving? And as it turns out, what's the key is not an insect, but the ohia tree, which is both culturally very important in Hawaii and very important to Hawaiian cave life, because these things are very good at colonizing lava flows once they've cooled down enough. And so their roots actually reach down into the caves, and it's these that are providing the base resources for Hawaiian cave life. So rather than bats coming in and providing guano, you have these roots coming down through the roof and plant hoppers, which are sucking the juices out of that. And so everything is building on that base. And so once these first cave animals were found in Hawaii, it was really sort of a huge surprise. And of course, more surveys were carried out. And in the last 50 years, surveys of over 60 lava tubes have found about 45 different cave species in Hawaii. So there is, in fact, quite a bit happening where we didn't expect there to be anything at all. And what we have are about 11 core taxa of cave-adapted life in Hawaii, including plant hoppers, assassin bugs, water treaders, which are really interesting because there's not really water to go around in Hawaiian caves, so they're cave treaders, wolf spiders, crickets. And so what this body of surveys has generated is our Hawaiian Cave Arthropod Collection, or HCAC for short. And that's about 40,000 specimen lots, almost entirely fluid of cave life. And of course, this is the largest collection of Hawaiian cave life. And many of these things are rare or endangered. We'll talk a little bit about their population sizes in a bit, but this is a really unique collection, not only for its size, but because a lot of these specimens are probably irreplaceable. We can talk a little bit about some of the cool things which are found in Hawaiian caves. So we've got wolf spiders, which outside of caves are known for their really large eyes and fantastic predators because of it. But this is Howarth's cave wolf spider, also known as the little-eyed spider, because of course, in caves, those eyes aren't doing much good, so they've degenerated and produced a non-visual predator, which can't actually match this other wolf spider, the cow-eye cave wolf spider, which is also known as the no-eyed big-eyed spider because it's lost its eyes entirely. So a bit of a strange fate and a strange name, but it seems to be doing just fine eating this thing, the cow-eye cave amphipod, which of course is blind and pale and doing the typical cave adapted life thing. And all of these species are federally endangered. So they've been formally designated as endangered to reflect their small population sizes and the very fragile environments in which they live. Speaking of fragile, we also have our collection. I'm very fortunate that this talk was after the excellent talk on fluid preservation, because that is a major issue for our collection. So the first major issue facing our collection is just that there's been a long pause in curation following some restructuring at the Bishop Museum. So we had this excellent collection, but it was sort of sitting fallow in our collection with fluid evaporating. And also a lot of it was collected and hasn't been fully identified. We have no inventory of the species in our collection as a whole. We also know that we have a lot of these specimens that went out on loan and haven't returned yet, and it's difficult to track where all of those are. So if anyone out here in the audience happens to have some of these specimens, please email me. We would love to get them back, to digitize them and then send them right back out so experts can ID them and all of those good things. So that sort of led us to our digitization project, which is funded through the Institute of Museum and Library Services Museums for America project. So we have three main goals. So to digitize and image our specimens, as well as our field notes. I'll talk more about those in a minute. To rehouse our specimens, both in terms of getting them into nice new vials and topped off with more ethanol where needed, as well as to make sure our specimens are as well organized and well identified as possible. And our ultimate goal is to really establish the HCAC as a cataloged resource, not just for entomology, but also for cave biology as a whole, because these really were an important development in the understanding of cave biology as a field. Finding 45 species of cave adapted life in an area where they weren't supposed to occur at all was a major advancement. Not that I'm a biospeleologist, if I'm even saying that properly. And they're also important for understanding endangered species in Hawaii, invasive species too, but that's for another talk. So we really want to secure our specimen condition as well as to establish a checklist of Hawaiian cave life. So to talk a little bit about our progress at the moment. So we've gone through and we've centralized our holdings. So previously these specimens were sort of scattered throughout our collection as well as in the personal collections of researchers. So we've really tried to centralize those as much as possible, barring the loans mentioned earlier, so that we can get those ready for digitization. So our digitization is still in the early stages. I don't have big numbers to show off today. But it's really helped us out to sort of go slowly after the pandemic to sort of start to troubleshoot and refine our digitization protocols, which has been nice. So one really exciting thing is that a lot of these specimens were collected by Frank Howarth. And so we have all of his field notes going back 40 years. And so a lot of the collection is covered by these field notes, and it includes not just where the specimens were found, but maps of the caves that they were in, rearing notes for when plant hoppers were pulled out of the cave and tried to survive in the lab. So it ties the specimens to a really critical environmental context, and it's a really exciting opportunity. To talk a little bit about the cave environments. So these are really sensitive environments, and that's both true in an ecological sense because they're fragile, very sensitive to disturbance. Species populations are small. The spiders and the amphipods that I mentioned earlier, whenever they've been surveyed, they've been less than 100 individuals at any given site, often far less. Some of these caves are federally protected. Others are on private land where people may or may not be willing to have them explored. And that's posing a major challenge for our collection digitization effort because a lot of our locality data is sensitive. So previously for the terrestrial parasite tracker, we were doing a lot of crowdsourcing for transcription, but that's something we can't necessarily do here because we can't be putting out these cave environments willy-nilly on the internet. Of course, we'd like to trust that no one's going to jump into a cave to try to collect the no-eyed, big-eyed spider, but that's posing a major challenge for us. But it's also a really exciting opportunity for us to demonstrate the importance of these fragile environments and make sure they're protected, as well as to provide outreach to a native Hawaiian public and visitors to the islands as well, who really appreciate these lava tubes for their beauty, but maybe don't necessarily yet know the cool, even if not so beautiful forms of life living in the caves. So really, just to sum up, our goal is to leave the animals in the dark, but to pull the information out into the light. So I'd like to acknowledge several people, especially Jim Boone, my predecessor, who this project is his brainchild, and he unfortunately passed away before it could be brought to completion. And thank you all. If I have time for questions, I'm happy to answer one or two.
Mike?
How is your reference cave step? The entrance, some point underground, depth?
So the question is how we georeference cave specimens. So I think that's still a process that for us is in flux. I think what makes the ideal situation there is if we have the map of the cave environments in these field notes, we may actually be able to point to a very specific area in the cave where these specimens were found. That may not be the case for all specimens, in which case the entrance is probably the clearest answer.
The other Mike.
Once you make all of these data on endangered species retrievable, what does that do to your record keeping and reporting to the feds on federally endangered specimens? Now you have tens of thousands of them. What do you have to do with that?
So the question is, once we have all this information on these federally endangered species, all these new localities or localities that are being pulled out digitally, how does that impact our reporting? And so I think the biggest change is going to be less looking back and more looking forward, so that there are surveys which are carried out on these specimens or these species every few years. And this will hopefully help us identify which caves are productive to look at. These are really highly localized species. It might be one lava tube has 16 of these spiders and another lava tube might not have anything at all. So it'll really help us identify which caves are important to keep looking at and to protect going forwards.
Follow up. There's got to be a federal agency that wants to know where every endangered specimen is carried out. What do you have to do with that? Everybody's got to know where every eagle is.
Sure, that's something I'd like to get back to you on. I'm still focusing on finding where all the spiders are in my very small office area. So the broader context will hopefully be for next year's talk. Thanks all.
Next up is Greg Brabant, and he's going to talk about the Wisconsin Insect Research Collection and the LabNet Penn grant they just received.
Great. I am the curator. I'm a hymenopterist who works for a coleopterist, and today I'm talking about Lepidoptera. Be kind. I also use social media. It's usually slow and clumsy, but here are the relevant details. I'm not going to talk about our physical collection, but if you're interested, I put together a video tour last year and I talked about how we transformed the collection from this to this. And so I would encourage you to seek out that video if you're interested in seeing how that happened. But instead, today I'm going to talk about our contribution to the Lepidoptera of North America Thematic Collections Network, the LepNet TCN project, which ended earlier this year. And a lot of you collaborated, were active participants, so I'm not going to talk a lot about it other than to say that initially there were 27 institutions contributing records. And the goal was to free up about 1.75 million occurrence records for butterflies and moths in North America, and also capture both low resolution and high resolution images for another 250,000 or so specimens. We applied for and were awarded a Partnerships and Enhancing Networks, or a Penn grant that started officially on paper in September of 2020. As you might remember, that was probably a poor time to start anything, but we did. And our goals were to add 70,000 additional records and take pictures of 1,500 exemplar species. So the initial group of collaborators represented the upper Midwest fairly well. We had institutions from Michigan, Ohio, Indiana, Minnesota, and even already from Wisconsin with a Milwaukee Public Museum. But like a lot of regional collections, we have a strength in regional representation of insects, including butterflies and moths. And in our case, that goes back nearly 150 years. And we also have good representation of less well-represented groups, like some of the smaller families, smaller in terms of physical size, and then smaller in terms of the number of species of micromoths. And we have good representation from habitats that maybe get overlooked, like bogs and fens in the upper Midwest. Again, like many of your collections, we periodically get nice specimen donations. Relevant to this talk, we have a nice underwing donation, and the Rocheleau collection is really nice because it comes from southeastern Wisconsin, which has seen an incredible amount of growth and habitat change over the last few decades. And I'll remind everybody that we were actually one of the original collaborators. NSF came back and said, we're funding you, but cut your budget 10%. And rather than cut everybody across the board, a few institutions, including us, dropped out of the initial project. In my Penn proposal, I talked about the importance of Wisconsin in the tension zone, in this case, Curtis's tension zone. This is a corridor that runs from northwest Minnesota down through Wisconsin, across lower Michigan and into Canada and New York. And it's best represented in Wisconsin, at least in terms of the length of tension zone. And it simply is this area that encompasses the range limits, in this case, of over 180 plants. And so the northernmost limit of southern species and the southernmost limit of northern species. And in the full title of LepNet, which I can't remember, it talks about the largest clade of herbivores, right? And so wouldn't it be nice to have baseline data for those herbivores, both north of and south of this tension zone? Because as we know, there's a lot of species of Lepidoptera that have narrow feeding preferences. Some are obligate on single species. And so having this data would be really important. Varies in width, and this tension zone is and will be moving north. And so having additional data about herbivores that might be feeding on plants provides some good starting data. So my methods were pretty straightforward. I wanted to compare the Lepidoptera records in SCAN. I didn't restrict the search to just LepNet partners, but basically any Lepidoptera records that are in SCAN. I did exclude observational records because we're adding to the specimen records, and I wanted this to be an apples to apples comparison. I also didn't want to project the or overemphasize the impact of our collection, and so I only included data today that represent specimens that have already been transcribed, georeferenced, and added to our database to specify, so they're ready to be shared with SCAN, iDigBio, and GBIF. And then I just created some maps using Google Earth and made a distinction between any record from SCAN versus records from Wisconsin. So I'm going to give you a series of examples that I hope emphasize the importance of including regional collections in some of these big studies. So the first family is Schreckensteinidae. I chose it honestly because I wanted the chance to say Schreckensteinidae in a presentation, but I also chose it because there's only, we only have 3 specimens in our collection, and they come from 2 sites. And out of context, that might seem like a pretty small contribution, but if we add in all of the records for Schreckensteinidae in SCAN, you can see that those 2 localities represent 2 additional states that are otherwise not represented in the scan data, Wisconsin and Minnesota. The next family has quite a bit more records in scan. And oh, I jumped the gun a little bit, but there is a very large Wisconsin-shaped void in the data mapped from scan. And when we add records, we certainly have a pretty good coverage in Wisconsin, but we're also submitting our records, all Lepidoptera records, not just those specimens from Wisconsin. So we add additional data from the upper Midwest. Glyphipterygii, that's a fun one as well. I zoomed out a little bit because this is, of course, all of North America. And, you know, there's good representation in scan, East Coast, West Coast, up into Northern Canada. But there is, again, this Wisconsin-shaped void in the data. That's kind of a theme. And we are going to be adding a decent number of records, both in Wisconsin, but then also in Michigan and Minnesota. So you might think that I'm cherry picking those families that don't have good representation in scan. So I switch over to the gracillariids and we zoom into Wisconsin, and certainly scan has some records from Wisconsin. So maybe our contribution for this family isn't going to be that significant, but if I add the records that we've databased so far, our sampling is a little bit more comprehensive, both in Wisconsin and across the upper Midwest. So this is in progress. We started again in September of 2020. Didn't really get started until the second year of the project, and continue that now into the beginning of our third year. You can read that, I'm sure, but 89 Lepidoptera families in North America, and almost a third of them were going to be at least doubling or greater the number of records from Wisconsin. And remember, my pool was 2.2 million Lepidoptera records in scan, so I think that's pretty cool. And for 6 of these families, again, the smaller micromoths, the ones that aren't well represented, there's going to be at least a 10-fold increase in the records from Wisconsin. So where are we? We're just over 29,000 records, so about 43% complete. We have the rest of the third year, and we will, I'm sure, request a no-cost extension, and we'll have no problems reaching our goal and actually exceeding it. I'm not going to talk a lot about imaging, but here are pictures of those 4. What is that? 12, 13? 5 minutes. Perfect. I deliberately left off the scale bars, but if I put them in, add some context for you to see the size, I hope that our contributions to high resolution images of some of these less well represented groups will be as significant as the occurrence data are. And then here's just a snapshot, which I think is kind of cool. You can probably guess, if you're not from around here, where Wisconsin is. And you can probably guess where some of our people that have donated material have gone on road trips. I'm going to take a moment to talk about the value of collection objects. And I hope this is in a little bit complementary to what Max so eloquently said before, just not so eloquently stated. But I want you to consider a collection object chosen completely at random, maybe this one. And when I have people come and visit the collection, I talk about 2 main components to the value of an insect specimen. Again, from my perspective, I'm simplifying, but there's this existential value. Sometimes I call it the inherent or the intrinsic value. And it's because that specimen exists, right? You can look at it, you can examine it, you can illustrate it, you can image it, you can measure it. And I just got done talking about the value, the scientific value. And so those 2 components, the existential value and the scientific value, again, in my opinion, are the 2 main components for specimens from a research collection like ours. And my responsibility as curator is to protect the former and disseminate the latter and advocate for both, right? But sometimes there's a third component, and this component is often of greater importance in other types of collections. You might imagine an art museum, and there's this aesthetic value. So I talked about the Rochelot donation before, and the first two components are well represented. They're very well curated specimens. They have very good scientific data. Everything's labeled. Most of it's identified to species. But there's also an aesthetic component to the value because of the way that he organized the specimens in his collection. And so we got about 100 drawers, and about 30 of them are organized like this. And I think you might agree with me that that can have a greater impact than showing the same drawers with the same specimens organized in a more traditional way. So if I have to suggest 2 takeaway points, I would say we have a unique assemblage of collection objects, just like every other natural history collection. That is to say, there's a lot of overlap, but there's never complete overlap. And we talk about natural history collections as archives of the species living on this planet. And so support for any single natural history collection, I think, should be celebrated in our community. That's it. Apparently I can answer one. I have time for one question. See if I can answer that. If you follow me on social media, I teased a recent acquisition of a 3D printer, and because of time, I'm not going to show anything. I'm sorry. Oh, I didn't say thanks. Thanks to everybody. But I will say that this was not bought with grant money, with any kind of grant money, and I can assure our director that it's only going to be used for the power of good.
Our next talk is by Torsten Dikow and his friends, and he's not here, and he's, I don't think, online. So if you have any questions in the program, his email address, so you can email him. And so enjoy the video.
It's a pleasure to be here today to talk about the digitization of the USNM Dragonfly and Damselfly collection. The Odonata collection at the Smithsonian National Museum of Natural History is one of the largest of its kind. With more than 3,200 species and more than 116,000 specimens, it's a large global collection of dragonflies and damselflies. On the top right, you can see a photo of how the enveloped specimens, which is the vast majority of the collection, are housed. And on the bottom, you see a map of our current data at GBIF that depicts the majority of these specimens. The Odonata collection here at the USNM is the only major insect taxon that has actually been databased at the specimen level. The data capture goes back to about 20 years, to 2001 to 2005. And 75% or a little more are actually georeferenced so far. The enveloped specimens, as you can see here on the top of the slide, are quite unique in the entomological community, but they sort of provide an opportunity to maybe use conveyor belt systems like herbarium conveyor systems to photograph them in a sort of more efficient way than a pinned insect in a three-dimensional way would be. And so we here at the Smithsonian decided that this would be a way forward to photograph every single specimen based on the pictorial herbarium conveyor belt that has been working in our museum for the past years to photograph about 4 million specimens of the US herbarium. The Digitization Program Office here at the Smithsonian has been working with Pictoria as a digitization partner for a few years. And so we decided to embark on this project. And the photo that I'm showing here on the top is from 2019, in which Pictoria staff photographed a few specimens just to get an idea of how this would work. This is a photo of 800 ppi that was the same resolution as the herbarium sheet, the herbarium conveyor system. And you can see already, we can see quite a bit of detail. But for this project, we decided to do an item-driven image fidelity analysis, which is an analysis technique developed here at the Smithsonian Digitization Program Office, to basically determine the smallest structure that we want to resolve in a photo. So I took two specimens, a damselfly and a dragonfly, and measured the CT on the head, on the legs, and in other parts of the body, as well as the wing veins, in order to find the smallest structure that we want to resolve. And the small structure is about 10 micrometers in diameter. And so we needed a resolution of at least 2100 PPI. And this very high resolution is still a compromise between the depth of field that the camera can achieve, or the lens can achieve, the lighting that is available, as well as the distance between sort of the specimen and the camera. The photography was done on a phase 1 100 megapixel camera back with an LED light source that was positioned at the top right of the specimen with a bounce back screen on the left, so there was even light across the envelope. And a single photo in RAW format was taken, which converted to a TIFF file is about 150 megapixels or megabytes large. You can see on the right-hand side just two photos and 200% zoom in into these 2100 PPI photos to appreciate the detail that we are going to photograph the specimens at. We decided to use a static setup and not a conveyor type setup because of this high resolution at which the specimens had to be photographed. And this is due because we wanted to avoid any movement, potential movement of the specimen. So here are a few photos of the imaging setup. You can see on the left our digitalization technicians, Nicole and Logan from Picture, who were handling every single specimen. Nicole took a specimen out of the box, scanned on the backside the USNM ENT identifier. And then Logan takes that specimen and puts it on the specimen stage, steps back, initiates the photographs, and moves the specimen into a different box, but in the same order from which the other one came. On the right-hand side, you can see the imaging stage, which can be adjusted in height, or basically the distance to the camera, and therefore the depth of field can be adjusted to from the skinny damselflies to sort of the chunky, thick dragonflies in order to keep everything in focus in that single shot. Nicole and Logan were able to have sort of an average daily throughput of 1,700 specimens, which a few days actually hitting 1,900 specimens and beyond. So quite a few specimens were photographed, and the whole project took about 17 weeks to actually photograph all specimens. The images were visually inspected right before Logan and Nicole moved on to the next specimen, just to make sure everything is in focus, is in view. And there were really no issues found with it. But there was also post-capture automated checkups of the data of the photos. And this would be checking the file name, then duplicates, the color, matching the color guide, or RAW and TIFF settings. But the final quality check was a visual inspection of 1 to 2% of each day's photos by Jessica Bird, who is our entomology data manager. And she said had the final say to say, okay, these photos are okay. Because, as you can see on the right, there are two photos. You can't see the actual USNM ENT identifier label in the photo because it's on the back side of the envelope and was only scanned in. We decided to use that information and put it digitally printed onto the front of the photo, if you wish. And so at the very bottom, you can see the USNM ENT number corresponding to that specimen. And that makes it easier for users to actually, cite this specimen correctly and have that information right there in the photo. The digitization cost was $1.04 per each specimen. And just like any other digitalization project in which you capture images of an entire collection, every single specimen was handled. And Nicole and Logan actually came across quite a few specimens that didn't have a USNM ENT label or number assigned to them, and therefore they were not in our EMU database. So we discovered these and then photographed them and added those into EMU following this project. And this was mostly specimens that had been identified only to the family level and not to species or genus so far. The way forward for us is to provide access to these images through our own data portal, through the Smithsonian Open Access data portal, as well as through GBIF. Images of Smithsonian specimens are in the public domain, have a CC0 license, and therefore can be used for scientific use and for anything else you can imagine doing with these photos. So we're looking forward to actually make them accessible. We have to put some resources into georeferencing because we have about only 75% georeference so far to put more dots on the map. And these will be mostly specimens from Brazil, China, Indonesia, India, and Sri Lanka. So we're working on that right now. We also would like to explore some machine learning applications, potentially for the identification of species or genera or families, and also to use that to make that image data set more widely known to the entomological community, but also to the scientific or machine learning community at large. Lessons learned for us is there was basically no damage to specimens. And as you can imagine, the specimens are surrounded by a card inside an envelope, so they're quite well protected already. However, there obviously was breakage before in envelopes, or when the specimens were remounted from pinned, relaxed, and put into envelopes. But these pieces are inside the envelope and have also been photographed. One of the points that we didn't consider at the time of development of this project early in the spring, and I'm not sure why I didn't think about that, would be to photograph the backside of the envelope if we do this project again. The vast majority of specimens will not have anything other than the USNM ENT label, but quite a few specimens will have the original labels that were attached to the pinned specimen, or maybe sort of the triangle envelope in which they were preserved in the field with some notes on it. So that information is not accessible from the envelope, but all the data are on the front, as you can see, on the front of the card, and therefore is visible, and obviously also captured in our INMO database. Excuse me. The images will soon be at GBIF, as well as the Smithsonian Open Access Portal, as I mentioned already. And finally, I just would like to acknowledge the funding that came from the Smithsonian Digitization Program Office, the Smithsonian National Collections Program, as well as the Natural History Museum itself. The digitization was primarily done by a Picture team, but Nicole and Logan, as well as management people. The DPO office, Digitization Program Office, helped with management. And Samantha and Julia, two contractors in botany, helped to move the specimens from the east side of the building where I am to the west side where the imaging setup was. And then collections management, obviously in entomology, it supported this project as well. Before I close, I would like to point out that the presentation PDFs and the recording will be available under this DOI for download as well. Thank you very much for your attention.
All right, we're going to start up in half a minute, so please settle down. All right, please, everyone back to their seats and quiet down. Unbelievable. You give them a little finger and then just... Right. Oh, we're getting there. We're getting there. Okay. I would have to know those. All right. So next up is Hannah Kernen. She is from Purdue University, and she's going to talk about the boiler bees. So please sit down.
Everybody ready? All right, I'll get started. Hey everyone, thanks for coming out. My name's Hannah. I'm a new master's student at Purdue, and today I'll be talking about our boiler bees, our bee collection at the Purdue Entomological Research Collection, also known as the PERC. Before I get started, I want to introduce myself real quick since I'm kind of new to the ECN scene, and there I am, right over there. But I mentioned I'm a new master's student. I study bees, obviously. I started this research during undergrad at Wellesley College in Dr. Heather Madela's lab. I worked at her bee lab for a few years. Upon graduating in 2020, unfortunately, I found myself as a research assistant at the Harvard MCZ working with Crystal Meyer. And at the MCZ, I worked on the big BTCN, which they'll talk a lot more later at ESA. But it was here I started learning more about digitization, thinking I really wanted to integrate this into some future grad studies. And I was lucky to find the opportunity to do so at Purdue University, where right now I'm working on a thesis on the bumblebees of Indiana. And I'll talk more about that in a bit. For the presentation today, I'll be hitting on a few topics. First, I'll talk about the history of the PERC bee collection starting from the early 1900s. Then I'll touch on some current thesis work that I've been doing, what we've done so far in relation to that project, and then propose a few directions that this research may take in the next few years. So to begin with the early years of the PERC, that begins in 1912 upon the establishment of our Department of Entomology when we split from the horticulture department. We got our first department head 8 years later in 1920, J.J. Davis. You can see a great photo of him down at the center there. J.J. Davis served for over 35 years as department head. He enacted a lot of positive change in the department, including in his first year, the official creation of our research collection. We had a teaching collection up to this point, but it wasn't until 1920 that it became a research collection. There's a great photo. All these historic photos were scanned by Chris, by the way, from our archive. That's a real photo on the left of Purdue students using this teaching collection. But even though we had our research collection, it wouldn't be until 1959 that we got our first full-time curator, Dr. Leblanc, and I'll talk a bit about him more later, because it's during this period from the 1950s to the 1960s, it's our first time of major growth in our bee collection, largely due to the efforts of two researchers who I'll mention today, Basil Montgomery and Leblanc. So I'll begin with Basil Elwood Montgomery, or Monty, as he was more commonly known. Here's a nice photo of him in New Zealand in 1949, looking at a box of bees. Monty was an important figure in the early stage of the department. He was brought on as faculty in 1929. He served for almost 40 years. He's known as a classical scholar and an early authority on Odonata, which was his main area of research. But interestingly, as I saw in our departmental history book, you can see a clip from that at the bottom, also a bumblebee researcher, apparently. I thought this was interesting. I'd never read any publications by Monty on bumblebees, so I looked a little closer and hilariously found that this might be a bit of an exaggeration, and that in reality, according to Leblanc, Monty's interest was the Odonata. He didn't know one bumblebee from another before he went to New Zealand and never progressed much upon return, which I thought was a bit strong in wording. We did know that Monty took a sabbatical to New Zealand in 1949 to study bumblebees, but apparently seems like this was a bit of a bust because he never published anything about that after the fact. But not all for nothing, Monty was a very prolific collector. We have thousands of bees attributed to him at the PERC, and more importantly, several years later, published the Anthophilus insects of Indiana, which was the first and actually still the only published list of Indiana bees. Very important. Now moving on to Leblanc, who I've mentioned a few times already. Unfortunately, this was the only picture I could find of him. I mentioned he's our first curator of insects starting in 1959. He was also known as a World War II Marine vet, a sheep rancher on the side, and had a mutual dislike of dogs, which was a very interesting detail that I saw in this same history book, which I should note that Chandler himself had a hand in editing, so I can only assume that this description came through him first. So regardless, he was definitively our first bumblebee researcher at the PERC, and a very prolific one. He published the Bombidae of Indiana, the first list of Indiana bumblebees, a few identification guides, and an interesting observation on Leslie Goss and ticks on him. Also a prolific collector. We have tons of his bees at the PERC. Moving on from this period, from the 1970s to the 90s, we don't see much in terms of bee research, probably because we were a bit busy building our ethanol collection, and Chris will be happy to tell you all about that if you have questions. But sticking to the topic of bees, we begin to see interest pick up again through the 2000s and the 2010s. This is in line with a greater national interest in bee decline, sort of the Save the Bees era, if you will. And at this time, we see the use of our collection in large-scale bee research. For example, on the top right, we have hundreds of bees identified by Dr. Jennifer Grixty for use in Cameron et al.'s groundbreaking paper, Patterns of Widespread Bee Declines in North America. Additionally, we see our first digitizing and imaging efforts in the collection at this time, mainly through the efforts of two local experts, Liz Day and Robert Jean. You can see some of their specimens there as well. And before I move on, I want to touch on Liz Day's efforts a bit more. She was a tireless and very interesting researcher in bumblebees. I heard she was a very fun character to speak to. She was very interested in bumblebees and birds and contributed hundreds of beautiful specimens to the PERC, including this wonderful Bombus affinis here on the left. This specimen in particular was prepared specifically for imaging for an unpublished guide to Indiana bumblebees, which unfortunately she was unable to finish before she unexpectedly passed in 2015. However, I am hoping as part of my work here at the PERC, we'll be able to catalog her specimens for the first time. And that brings me to today, to the bumblebee thesis that I mentioned, in which I'm hoping to answer the question, how have our Indiana bombus changed throughout history? And the ways that I'm hoping to look at this are through changes in time and space, through phenological and range data, respectively. And my overarching goal is to use digitized specimen records to track changes in species over time. So to begin, I started at home. I looked at our PERC collecting record. We have about 45,000 total bees at the PERC, and that includes 5,000 to 6,000 Indiana bombus, which is the focus of my research, of course. But looking at the distribution of these records, they're not super complete. We have a large cluster of samples around Tippecanoe County, where Purdue is located, of course, in the adjacent counties in that central western region. We also have a nice cluster down in the far southwest in Posey County, where Purdue has had a research station for several decades. And throughout time, the clustering is as expected throughout that 1950s and 1960s period with a bit of older historic material. And that star is just to denote that we do have modern collections from the 2000s on, but we haven't had the chance to catalog them yet. And so these gaps indicate a need to build our database to fill out those undersampled areas of the state and also through time. And there are three ways that I'm going about doing this for my project. The first is filling out records that we already have. And it turns out we do actually have a lot at Purdue sitting around Smith Hall that have never been touched. Primarily from cataloging from former and ongoing research projects. For example, in my lab, just in Ian Kaplan's lab, we have two other students, members of the lab, Allison and Ella, who have been collecting bees for years as part of their projects, and I'm hoping that their Bombas records will be digitized as part of this final data set. Additionally, of course, lots of material has already been published on bumblebees throughout the country, and looking at these digitized material will be important for building up this final database, though understanding you do not want to fall in the trap of uncritically scraping public records that exist, instead focusing on interrogating those records and finding the most important collecting events that will be helpful for our future data analysis. And then finally, to continue finding these unique collecting events, visiting nearby institutions to examine their undetermined material to see what hasn't been examined before. We've already had a great visit to INHS, and later in the semester, we're planning to go to the University of Kentucky and Hanover College to see material from those undersampled areas of the state. I also plan to go to the Indianapolis Children's Museum, whose insect collection has not been cataloged before, and also private collections, most notably Robert Jean, who I mentioned once before. Rob has been wonderful. He's been collecting bombas through Indiana for decades now, and he's been a great help on this project so far. And so I'll talk a bit about the early progress and challenges that we've had so far at the park. The first part of this was finding those stray bees, turns out several thousand that were just sitting in Smith Hall uncatalogued, and addressing any incomplete and degrading labels those specimens may have. And you can see two very great examples of both of those here. These are students who, the former on top did very well in all the content of his labels, but not with the actual material. They've become almost illegible after less than 5 years, unfortunately. And another student who, while his labels have stood up, they weren't exactly complete. So we've made sure to add new labels and voucher numbers with barcodes whenever necessary. And then also recuration. So far, I've done a thorough recuration of our Bombas specimens. And although these steps did take a lot of time, they were important in preparing us for the next step, which is digitization itself. And so throughout these previous steps, including accessioning over 200 of Liz Day's beautiful bumblebees, we have at least 1,800 specimens to digitize from the start. We've been assessing the previous digitization work that has been done, but unfortunately I found that throughout the decades, little errors have begun to compound, and that there is a need to overhaul these records anew and to revisit all of our bumblebee specimens. And so if we're going to be correcting and completing all these existing records, we might as well get great photos of our bumblebees while we're at it if we're going to be handling them again. And so for this, Chris and I have been prototyping an imaging system. It uses 2 daylight LEDs and a 100mm lens, and so far has been getting us pretty good resolution with the labels, definitely good enough for transcription, and pretty good resolution on the bee itself. So we're excited about this, and we're hoping to get started with this ideally at the end of the semester. Additionally, we're hoping to take some high quality photos for representative species. I do want to be clear, I know this isn't a bumblebee. This is a great osmio specimen that I found and took a great test image of this setup. And we're hoping that these high quality images will be great for use within the park, for outreach, and for beyond. And also in the use of some final products for this project, which I'll talk about now. I'm in the early stages of my thesis, so these are a bit in flux, but these are the thoughts that I have so far. Without getting into too much of the details, the grant that I'm on emphasizes the development of a potential online resource, which in this case I think would be a guide for Indiana bumblebees for use by the public. And I feel that a project such as mine can contribute very importantly in a few interesting ways, particularly in the use of visual data. And for example, through contributing high quality images, range maps, charts showing phenological information could all be very useful for people who want to learn more about their local bee communities. Additionally, I hope to incorporate additional factors into my analysis, such as land use and climate variables, to learn a bit more about what may be contributing to these different changes in population dynamics over time. And then lastly, I'm hoping that the work I do can contribute to the infrastructure for digitization at the park. And so that even after I'm gone in a few years, future bee researchers can build upon what we've done and continue answering questions using our specimens. That's all I have for today. Thank you for my collaborators at Purdue and beyond. A special shout out to Tommy for a great visit, and I'll take any questions that you have. Time for one or two. Yep.
This day, I worked with her quite a bit. She was the one.
Yeah.
A lot of notes, a lot of drawings, manuscripts.
Yeah.
That I was doing. Did all of that stuff survive? All of her manuscripts and drawings and stuff, maybe salvaged?
Yeah, I know Chris will know more about this. We do have some of those drawings. The picture that I have, for example, this scan. Just want to show this up again. Okay. Sorry. Yeah, this was scanned from some of her unpublished work. And I know we do have a fair bit of it. We have collaborations between her and Arwen Provancha, a former curator. But I think the exact extent you could ask Chris, maybe Aaron would know as well. But we do have several hundred of her specimens. I'm unsure if that's all of them, but the ones we do have are beautiful. They're very well prepared. So thanks so much.
Go ahead.
Okay. Just a couple quick housekeeping things. We know we're out of water and coffee, but there'll be more after lunch. And number two, our wonderful treasurer, Patrick, has made it so we all have Wi-Fi. The password is all caps ECN, but there is only enough for 100 devices. So if everybody signs in on their phones and their laptops, it will crash. So don't do that. It'll get us in trouble. So just sign in like if you have to, and just one device, please, per person. Thank you.
All right, next up is Michael Ferro, and he's going to talk about libraries old and new.
Huh?
10 minutes is the yellow.
Oh, okay. So first of all, thank you to everybody who organized this thing. This is the greatest meeting in the world. I mean, honestly, think about the people who are here. The other thing is I have to give a shout out to Max Barclay because he, I mean, of all of the wonderful things that he said, he really, hit the nail on the head with kind of that. You get this palpable sense from Lord of the Rings of the loss of the old ways, and this is kind of a talk about that. There are some old ways that need to be chucked out, and there are other old ways that need to be kept alive. And please don't think that this is kind of a grumpy old man talk. I maintain every paper that I use as a digital copy. I scan in books. I do everything. And then I also have a giant, very heavy print library as well. So anyway.
You have space.
I have file cabinets on rollers and they're in the hall. They're in front of other things. You have to move them out of the way in order to get into places. I have space, but it's a dance. Okay, because every design must satisfy competing objectives. There necessarily has to be compromise among, if not complete exclusion of, some of those objectives in order to meet that consideration, what are considered the more important of them. And so there's no best design, there's no only design. Everything is a question of costs and benefits. There's no best mousetrap. And I think this is the best and only way to think about that particular issue. So this is not a paper about, or this is not a talk about paper versus electronic. This is, let's talk about costs and benefits. And this isn't a professional talk either. I'm just going to tell you about some of the things that I've run into recently. So one thing to consider as you're chucking out paper, or if you're going to be using electronic papers that have been scanned in, if it wasn't born digitally, that paper, when it comes to the image, any images that are on paper, that original image is the best we've got. That's the highest quality and that's the least distortion. You can almost think about it as the holotype. And so also appreciate that in most cases, those images, the original images, were turned into dots. They were printed. When you scan them or photograph them, they're turned into a different set of dots. When you project them or when you print them again, they're turned into a different set of dots. And so all of this translation can cause all sorts of issues. And so the general term for this is aliasing. It's when different signals become indistinguishable. There's an enormous amount of math behind that. Within images, we're familiar with the Moiré patterns when you have interference patterns. And That's amore. And so appreciate that if you're not already aware, there's lots of different things that you can use on Sharp Mask and descreening and other things to deal with color correction that exist in the digital world in order to take that image that has been altered and covered in artifacts and then run it through another system of artifacts that alter it again to make it less looking like it was. So, right. The point of this is that when you move from paper to digital, there's a human being in the middle. That human is either going to do something or not do something, and what they do or don't do matters. And if there's an idiot in the middle, you're not going to get a good product. And so let me give you an example. I had a tiger bee fly ovipositing outside my window during 2020. The eggs are less than 1 millimeter long. They're covered in dirt. So you don't know what you're looking at unless you know what you're looking for. I got online, I got a PDF of the 1976 paper, where they took SEMs of the eggs. That's the one on the far right or left. Anyway, that was the free version. My school has a paper copy of this paper, and I asked them to send it to me. And of course, they took a photograph of it and they shipped me that thing in the middle. That book, by the way, is 20 miles away in a warehouse, so it's not easy for me to get. I asked them to send me the actual book, and I scanned it in myself, this one that says My Scan. And so please appreciate that if we were looking at punctation on the pronotum of a cravid, you've got 3 different species here. Consider that. Now, you can buy this 1976 paper from the publisher for $40 if you want. But when you do that, what you get is you get the file that was already available online. So the worst scan is the official scan. Just want to point that out. The other thing, a brief side note here that I want to point out is that we think of the digital being wonderful because it allows us to copy with pure fidelity, but every PDF that's out there is going to be copied, and it's going to be reduced, and it's going to be altered and moved to a different format over and over and over. And so ultimately in the future, there will be hundreds or thousands of versions of the same PDFs, whether it was born digitally or not. And just keep that in mind. Another fun example, I wanted to use this image. It's out of copyright in a publication. You can get it for free on Biodiversity Heritage Library, the optimized version there. You see a little cutout. They do this in in Adobe. It looks horrible. You can download their original picture that they took. It's not print quality if you want to do that. I actually bought the rights to use this image through the Royal Society Picture Library. It cost me $50, and they sent a pretty good scan. I didn't particularly like it though, and I suddenly realized, what does the book cost? So I bought the book for $50, scanned it in myself, and now it's out of copyright. I don't have to worry about licensing or how it's used or any of those issues. The other nice thing about actually owning the book's full of tons of other illustrations and things that I can now use. The scan of the bee head there that's mine, that took less to scan in and clean up, less time, than it did to find in Biodiversity Heritage Library. And if you've ever tried to clean one of these images up, it is murder. And so having that in your pocket is just wonderful when you have the paper available. But don't just think about images though. I was doing this kind of global survey of all of the mentions of insect behavior during solar eclipses, and you run into stuff in Japanese from the 40s. It's not just that I don't read Japanese, that I can't read that. Cyrillic scans and OCRs and translates perfectly, except when the paper is warped. And so in these cases, I went to all the effort of asking for the paper interlibrary loan. What they sent me obviously doesn't work. I have to ask for it again. But foreign languages such as English also don't come through very well either. So this is Blatchley's. We've talked about Blatchley before. Here's Blatchley's Coleoptera of Indiana, 1,300 pages. There's the free version Biodiversity Heritage Library, the free version Google books, and everything. If they've been OCR'd, everything in yellow is correct. So if all you have is that electronic version, you're going to spend a lot of time trying to read that, which is stupid, or you're going to do searches, and your searches are going to turn out very poorly. So just consider we're going to need to keep that paper around at least a little longer until we do things correctly. Also, for the fun of it, born digitally as well. So I wanted to copy a material examined section for somebody. This is 2019 Coleopterus Bulletin. When I pasted it, everything that's yellow was a mistake, an error, if you will. And this isn't just a translation from Adobe to Word issue. If you copy the equals sign out of the Adobe and put it into the function, search function of Adobe and ask for it to search for it, it can't find it. And so there's all sorts of fun little shenanigans going on in the background. So just always keep this in mind. We've talked about, or we've heard the term value several times, and I'm very excited to hear that. Anyway, so what's the cost of free and electronic papers and books? Is there a cost even? We all appreciate free materials, Biodiversity Heritage Library, research-based. Sci-Hub moves around because it gets taken down. Google searches, journal websites, all that fun stuff. Is there a cost to this? And I would suggest that there is. It takes time. All of us have been in that situation where we find a paper we think might be interesting, then we gotta run a circuit before you can find an example of it so that you can maybe download it, or it's behind a paywall, you gotta log in, you gotta do all of this stuff. There is substantial time involved in obtaining this quote unquote free literature. And so we don't value our time because we like what we're doing. But if you hated what you were doing, by God, all right. So how much of your time do you spend finding a paper, downloading it, cleaning it up, working with it? Interlibrary loan, the standard is they say $17.50. But estimates think that it might be $35 to get an item through interlibrary loan. So I would suggest the printed versions save time. I would suggest that, here's an example. I did a paper updating state level distributions for Coleopterigidae. I downloaded, I got every paper I could, printed them all off, etc. Used them. 4 years later, somebody, Al Wheeler, found some in grasses. Nobody's ever reported them from grasses, or have they? 45 seconds to walk across the hall. I can immediately start going through papers. I immediately start building a huge table on what's what they're known from. So anyway, and you've done this as well. So when you think about a library, a reprint collection, what is it worth? So that paper that was printed off from a PDF, that you're not going to sell that on eBay. What is it actually worth? Don't worry about replacement. Don't worry about value. Just time saving, just basic time saving. If you imagine that each piece that you have is worth $3, walk over and pull a book off the shelf. That means that the file cabinet that I have on a cart is worth about $4,200 worth of time saving, if you will, value. And I know value sounds like BS, but that's all economics is BS. So anyway, just think about that. A few other things to think about when it comes to difference between electronic versus paper. Search biases. If you're working on phylogenetics of a particular group, do not go to the library, go online. If you're doing anything else with a particular group, go to the library first. What you get from that older material or from that other material is papers that other researchers found relevant. They filtered the world and then compiled it into a series of file folders just for you. Search versus discovery is very important. We have magnificent search engines. We do not have good discovery engines. The ability to find that thing that you don't even know existed is the 400-page book on the history of the pencil. You know, when that's what you see as you're going to the entomology section in the used bookstore. Field guides are spectacular for search and discovery. Discovery, all the moths that are available. Search, you flip through it and it takes a few seconds, much, much faster than even iNaturalist or Bug Guide. Sharing is caring. Your physical collection can be shared publicly. Everything from Henry Towns is available in my library or in CUAC's library. You can show up, you can look through it anytime you want. The problem is though, is that you have to be there. But if you have a digital library, we don't have the ability yet to be able to share that publicly either online. You're going to run into all sorts of issues. And so for those of you that have giant digital libraries, think about what's going to happen with that when you aren't around anymore. And I don't have any advice on this. What are we doing? We're adding our books. We think we have about 1,000 books. We're adding those to Library Thing. I went through and photographed every one of the reprints that we have in our collection and put those online. So you can go online and you can look through our reprint collection. Think about how amazing it would be to be able to look through other people's reprint collections. So consider doing that. What does the future bring? There's all sorts of possible issues that we might run into, restricted use of PDFs. We might start getting charged for downloading PDFs or interlibrary to the library, sunset dates on PDFs. Someday we may have to prove that we had access to the papers that we're citing in our publications. You think that's crazy, but we have to prove now that we had the right to collect a bug. Think about that. Try to convince somebody of that 100 years ago. We're going to have issues with multiple versions of the same paper. And as things move more and more electronically, we're going to have all sorts of fun shenanigans when people try to change dates, change names, and create fake documents in order to make it look like they did more than they actually did. And so I think that's, oh, please consider your library as a unique living collection. Am I completely out of time? 1 minute, there you go. Read that last line. Literature collections represent a unique assemblage of material, filtered and found useful, compiled by generations of researchers. If you allow it to grow and you continue to use it, it will stay alive. It is definitely one of the old things that we need to keep. Support your literature collection. If you don't want it, I'll take it. I will take it. And keep it alive. And thank you to Crystal Meyer for allowing this presentation to occur. Thank you very much.
All right, last talk before the break. So also last talk for this general section. We're going to have Luc Leblanc talking about 10 years of fruitful collaboration with his friends in Bangladesh.
Bienvenue au Canada pour nos voisins américains. Moi, je suis de la minorité francophone du Québec. So Canada is a bilingual country. In the United States, I'm part of the Missionaries Diversity and Inclusion. Here, I'm part of one of the two official languages in this country. But and Henry Towns, I used to know him. My initial career was as aspiring career was as economic taxonomist. So I spent lots of time with him. But today I'm going to talk about field work in Bangladesh. So I'm holding the Tales from the Field flag this year, and I hope that this series will be back very soon. So Bangladesh, so I work on tropical fruit flies, tribe Desaini, which includes over 1,000 species, including 83 major pests of fruits. So that's how I can get funding from USDA to survey the species that invade the United States in their countries of origin. And as part of that, started doing some work over the last 10 years in Bangladesh. Well, just to break a few stereotypes, Bangladesh has reached food self-sufficiency in the last 10 years. 90% are Muslim, yet the prime minister and head of oppositions are two women, and they have zero tolerance for fundamentalists. And they have a third gender called Hira, which is as close to recognizing transgender people as you can do. And Bangladesh has done it years ago. So it's quite a fascinating country with extraordinarily friendly and genuine people. So the Bangladesh Atomic Energy Research Establishment has an insect biotechnology division because of SIT, so they had support from IAEA and for their research. And you'll notice many women among the scientists on the photo below. So a lot of highly educated women, and this is the team I've been working with. Aftab Hossain on the left photo on the right side. He's entomologist, fruit fly entomologist. I received an email 10 years ago out of the blue about a Bangladesh entomologist who wanted to have some fruit flies identified, and I contacted AFTAB, and since that time to this day, we're still collaborating together. So below is the mass rearing facility for fruit flies and parasitoids for area-wide control. So the first 2 years, they did initial surveys and mailed to me the samples, and I identified the fruit flies and was able to publish with AFTAB as co-author, of course, 2 publications reporting on the fruit flies of Bangladesh, which increased the number of species to that increased number of species, because fruit flies had never been surveyed in Bangladesh until AFTAB started a decade ago. And then because of these publications, I was able to obtain some Farm Bill funding to go and survey pest fruit flies that keep appearing in California, in Bangladesh, and in Nepal. So I did 2 trips to Bangladesh. And as part of that, we started surveying the protected forest areas of Bangladesh. The middle map shows it, and the map to the right shows all the sites that have been surveyed so far. And there are 26 protected areas in Bangladesh. And one example, Lawachora National Park. So you think that I just arrive in Dhaka and then go there right away? No, you have to go through an entire day of impossible traffic jams and very dangerous driving. I was going to put a little film clip of that, but that would be for an hour-long presentations. And we reached Lawachora National Park and put series of monitoring traps for fruit flies. And of course, land leeches are ubiquitous in Bangladesh. And we cross river by because in the water, because the bridges have crumbled over the years. It's not a very rich country, but it's still really great. Rema Kalenga Wildlife Sanctuary. Again, we drive through countryside, through rice paddy fields. You see every square meter of available in Bangladesh that hasn't been flooded yet by rising sea level is all used for food production, except the protected areas, which they do protect. So, but then the roads, you eventually have to abandon the vehicle because the road is muddy, especially during monsoon season. And then we continued with tuk-tuk, and we had to get off the tuk-tuk when they would drive through mud. So it can take like half a day just to reach an area. And so that's Rema Kalenga Wildlife Sanctuary, beautiful park. And in Bangladesh, you don't see any tourists in these parks. You don't see any foreigners. You don't see any Westerners. In my 2 trips in Bangladesh, 3 weeks each of these trips, I saw one other Westerner who each trip in Dhaka, who obviously, like me, was on a mission. So it's a pristine country in terms of not having been spoiled by tourism. So the Sundarbans, this is the most extensive protected mangrove in the world, and it's a sanctuary for Bengal tigers. So we did some surveys, not many fruit flies because it's not the right habitat, but we still did some surveys around there. And on the top photos, you can see what our countries are doing to developing countries with rising sea levels. I've lived 21 years in the South Pacific Islands. I've seen it there too. So we have a lot of work to do. So Chittagong Hills, Chattogram, these are mountain ranges next to Myanmar, and they're the most extensive forests in Bangladesh. So we did go and did a little bit of trapping at the edges at where the forest starts, but we could not go into the forest because it's not, foreigners are not allowed. I'll tell you why afterwards. So Cox's Bazar, Chunati, so these are all areas that are in the southeast Bangladesh, at the whole region adjacent to Myanmar. So I was able to go there. Pretty unique. And oh yeah, funny, these two fellows on the right, to tell you that they don't have these, that it's a non-tourist country. The older man, he saw me and he just came because I was the only Westerner in the town and in the whole beach. So he came to me and said, "Oh, what is your country?" And I said, "I'm from Canada." And he went, "Oh, Canada is my favorite country of all." So I'm used in other countries of con men just praising you so they can ask for something. So I was thinking, so what does he want? And he said, "Yes, my favorite country. In 1970, when we had the war to become independent and become our own nations, the entire world ignored us. But your country, Canada, was the only one who helped us. So that's why it's my favorite country." So you have these genuine interactions with Bangladesh people every day. So another thing that attracts me about going to Asia is to go and to Hindu and Buddhist areas. I've been in many different countries, and these, so in Nepal or China, you go and or Thailand, there's big crowds, busloads of tourists. Bangladesh, you go to the local Buddhist areas, not a single visitor, it's all local. So this area I was able to, and including this giant Buddha statue you see there. So I was able to spend a lot of time in those areas and talk with the lamas and had really great time. So it was really amazing. So yeah, Bangladesh, it's completely untouched by tourism and it's amazing. And this area here, I would not be able to go back. This is, so the photo at the bottom, that's the border with Myanmar. So I went all the way to the border. I met a few Rohingyas who were refugees. And this was 1 month, July 2017, 1 month before the Rohingya crisis. So I was able to go to this area. Nowadays, you need a good reason and permits to go there if you want to go there. Likewise, the Chittagong Hills, they do not allow foreigners unless into the forest unless you apply for permits and you have a police escort, because they had a 20-year conflict with minority people in the Chittagong Hills. That it's safe, but they do not allow foreigners to go there because there was instances of kidnapping for ransom, just like with the FARC in Colombia. So Bangladesh, the last thing they want, there's so few foreigners going there, the last thing they want is newspaper titles. Canadian is adopted in Bangladesh. So that's the biggest regret I had, was that we were not able to thoroughly survey the most pristine parts of Bangladesh. So in total, we collected to this day 34 species of day-sign fruit flies. And that was published, described the new species, and did quite a few publications. And we did 2 years of monitoring of population abundance cycles to in preparation for area-wide control. And I delivered a training course on fruit fly identifications in 2017. We've published 5 papers out of these 10 years of collaboration. So it's really great. And right now, the current project, AFTAB is collecting in the field the other tephrites, non-designs, and Alan Norbaum is processing and identifying the samples, and he's been rearing larvae of species of design fruit flies, and Gary Steck in Florida. So Alan and Gary are my 2 fruit fly buddies in the United States, and I brought them into that collaboration. So there's a great potential for project and collaboration in Bangladesh, but you have to think about how to pay back and how they should also benefit by publishing everything, training people, and then you can have a really great collaboration. AFTAB got an award in 2021 for his work, which is really great. And just one slide. Last year, I sent a series of emails to ECN on a purple orb on the left photo. That's artifact, unwanted artifact from Olympus TG4. The right photo on the right side, that's with Olympus TG6. That problem was resolved, and it's a very affordable camera that has a focus stacking function. And Emma Ekens on the bottom left, she's by museum worker a few hours a week. As I said earlier, we in smaller universities, we have 1-person teams doing everything, including teaching. So I put her on a project to photograph every one of the buprestids and digitize all the specimens from Idaho. And she did an incredible job. The photo on the right, there was a pin through it. There were dirt on the specimens. Through Photoshop, she cleaned everything. She's Emma Ekens. I bet we will see her in the meetings in the future. She's a second-year undergrad. She's phenomenal. We have these incredible undergrad students, very passionate, and she produced poster on the jewel beetles of Idaho, which you can download your PDF on the museum website. So thank you very much for your attention.
We have time for maybe one question.
Yes.
I was just gonna ask, for these ongoing surveys, where are the specimens in Torsten? Are they split up between institutions or?
The majority is at the University of Hawaii Insect Museum because they're doing the molecular work. A subset is at the University of Idaho, and there's synoptic collections of pin specimens in Bangladesh. Yeah. And Bangladesh is great, unlike India and Nepal, which I came to realize. Bangladesh have minimal permit requirements. You can go collect. You just have to have local contacts, and you don't need to apply for these export permits and all that. It's one of the last countries in Asia, but getting there, not easy, and the day-to-day work of being there, it's pretty hard. But I have 5 years in some of the really tough African countries. You heard my talk on living through a genocide 3 years ago. And by the way, I was in Australia in July, and in the Queensland TAF collections to do fruit fly work. And I was able to go back and see my fellow genocide survivors who live in Tanzania. I was who I found after 16 years after they were dead. So I was able to see them again. It was fantastic. Okay, thank you.
All right, that was the last talk of our first general symposium. Thank you very much for participating, and We have a about 90-minute break